Good morning, students. We shall start with the next chapter in ethology. In this session, we'll be doing about pheromones. What do you mean by pheromones? What are the different types of pheromones? And you can see examples in insects and vertebrates which secrete pheromones. What are pheromones? In the year 1975, Novak described them as chemical messengers. These pheromones are chemical substances which are released into the external environment. It is released by an organism which is called as a releaser, and it brings about a change, or the message which is released along with the pheromone is picked up by another member of the same species. So it is referred to as intraspecific or species specific. One organism is releasing a pheromone, which is a chemical substance which carries a message, and this will affect another organism. When you say it affects another organism, it brings about change either in the behavior of the second one, or in the process of development, or in the process of reproduction. So one is releasing the chemical substance pheromone which brings about change in the other organism in its behavior or development or reproduction. So when we say the pheromone is giving out some message, what message is that chemical giving or passing on to another member? It provides information like the sex of the organism which is releasing the pheromone, whether it's a male or a female, or it can also give information about the reproductive status of the individual, whether it is reproductively mature or not. Now all of you know about hormones. Hormones are also called as chemical messengers, but hormones are released inside the body and it brings about some change within the same organism. So the hormone released in one part of the body is carried to another part which is called as the target organ, and this organ will bring about some physiological changes like release of a enzyme or release of a digestive juice or anything. It brings about change in the physiological responses of the same organism. So hormones are chemical messengers which are bringing about change in the organism in which it is released, whereas pheromones are released into the external environment and it brings about change in another organism. So the pheromones are released by exocrine glands. They have duct, so along with the duct the secretions will be released. In the year 1959, Carlson and Lucia described about the pheromones. The term pheromone is derived from two Greek words, pherein meaning carry and harmon meaning to excite. So pheromones are hormone-like substances secreted by exocrine glands released into the external environment. So it is called as ectohormones. As it is released outside, it's called as ectohormones. They will act like chemical messengers. They carry signals, and these signals in turn brings about responses in the members of the same species. So you call this response as intraspecific communication, wherein the release is trying to communicate some information with the receiver. So the one which is releasing the pheromone is the releaser, and the one in which it brings about change is the receiver. So there is a communication between the releaser and the receiver, which we call it as intraspecific communication. There are different types of chemicals which act as signals. So let us say there is a chemical which is bringing about a communication between two different species. So earlier we spoke about the chemicals which is bringing about change within the same species, that is intraspecific or conspecific. Here we are talking about chemicals which are bringing about changes or responses in two different species, then it is called as interspecific. So there are certain chemicals which are capable of bringing about changes between two different species. So the first type of such a chemical is called as alamone. In this type, there is an organism which is emitting. So the emitter which is releasing the pheromone is benefited. So the pheromone is favoring the emitter. So let us say there is an organism. It has got threat of being eaten by the insect. It releases the alamone. This alamone will act as a repellent of the insect. So it is protected by being eaten up by the insect. So it is between two different species and it is called as alamone. The second type of chemical is called as chiromone. Here it favors the receiver. An organism is secreting the pheromone which is received by another one, and the receiver will actually get benefited. For example, if you look at a parasite cannot survive on its own. It needs a host. It is always in search of host. Now this parasite can recognize some chemicals secreted by the host which will attract the parasite towards the host. So the host is secreting chemicals for some other reason, whereas the parasite is getting benefited by identifying that chemical, and it gets attracted and it gets attached to the host. Such chemicals are called as pheromones. So now there are different types of pheromones. Basically there are three types. The first type is called as releaser or signaling pheromone. So this pheromone is released by one organism. It follows a pathway which involves central nervous system, or you can say it as neurohumoral pathway. So the pheromone is picked up by the sensory cells. It is taken to the central nervous system. Then it reaches the blood. It follows the neurohumoral pathway, and an organism will release a pheromone which will act as a signaling chemical, and this signaling chemical will be perceived by another organism, and the other one will know information about the first one, which is the releaser. The first one is releasing the pheromone which is carrying some signal which is perceived by the receiver. So the first one, when it is releasing the pheromone, it gives information about itself, like the sex of the individual, whether it's male or female, the species, the sexual status, whether it is reproductively active or whether it is taking care of the young one. So this type of pheromone is called as releaser pheromone or signaling pheromone, which is released by a releaser, picked up by a receiver. It is picked up by the sensory cells, then it follows neurohumoral pathway and brings about responses in the receiver. Aggressive behavior which is seen in female. Suppose a female has given birth to young ones and a male approaches this female, or a different male which is not its mate approaches this female, she shows aggressive behavior. She releases a pheromone called a sex pheromone which will try to chase away the male which is not familiar to the female, which tries to come near this female or encounter this female. This is called as aggressive behavior, and it releases the pheromone, sex pheromone. Another classical example what you see in the animal world is the alarm pheromones, which are secreted by some insects. Alarm pheromones are those chemicals which are responsible to raise an alarm when there is threat to the colony. It is seen especially in those insects which have got social life where they live in colonies. To name a few colonial social insects, we have ants, honeybees and wasps. All of you know that if you try to disturb a honeybee colony, then one of them will come and sting you, and when they sting, they release alarm pheromone. This alarm pheromone will be picked up by other members of the colony, and they all try to attack you. How did they get the information that there is threat to the colony? When the first one bites you, it is releasing alarm pheromone which attracts other members of the colony, and their main goal is to protect the colony and chase the predator away. So this is again another example for releasing or signaling pheromone which is exhibited by the colonial social insects. The second type of pheromone is called as primer pheromone. Unlike the first one, the first one releaser pheromone, the second one is the primer pheromone. This one will bring about long-term effect in the receiver. It acts on the endocrine glands and brings about physiological responses in the body. Let us see an example. If you look at the rat reproductive cycle, they show estrous cycle like what is there in the human species, menstrual cycle. The estrous cycle in the rats, suppose there is a cage in which only female cycles are there, and these females will be releasing certain pheromones. Because of the effect of the pheromones, the estrous cycles become irregular. Now, if you introduce a male releases some pheromones, and these pheromones will act on the reproductive organs of the female, and it regularizes the reproductive cycle of the female, and it normalizes the cycle. So the female reproductive cycle becomes normal, and it is ready to mate the male. So in this case, the pheromones released by the male is acting on the reproductive glands of the female, and it is regularizing the estrous cycle of the female so that they are ready to mate. So the pheromone is bringing about a long-term change on the endocrine glands, bringing about physiological responses. So this is one example for primer pheromone. There was another experiment conducted by a scientist, Bruce, which is called as Bruce effect. In this experiment, when a female rat which is pregnant, which is being kept in a cage, now you remove the male which was responsible for the pregnancy of this female, you remove that male and introduce a new male into the cage. When a new male was introduced into the cage, then what happened to this female? She induced abortion and she aborted the developing pups. After abortion, she was ready to mate the new male. This was studied by Bruce and is called as Bruce effect. Why did the female do so? The female induced abortion of her own pups because if she had waited for the completion of the gestation period and if she had reproduced, then the new male who did not father these pups would have eaten off the pups so that he can mate the female and produce offsprings of his own. So the female knew this behavior of the male and hence she induced abortion. Anyway, these pups will be killed by the new male, so she induced abortion and she became ready to mate with the new male so that the progeny will survive. This is another example for primer pheromone. If you look at honeybee leads a colonial life. In the colony, there is a single reproductively active female called as the queen. This queen, when she is there, she will not allow other females to become the queen. She suppresses the development of reproductive organs of other females by producing queen substance. This is a pheromone which is released by the queen bee called as queen substance, which suppresses the development of the reproductive organs of other female members of the colony. Similarly, there is also social pheromone released by the termites. So these things we will be doing in detail when we do social life in the next chapter. The third type of pheromones is called as imprinting pheromones. So this is very important, like what we studied in the previous chapter. Imprinting is a critical state where the mother will bind with the offsprings when they are born, because the offsprings need to be taken care of. They have to be fed and protected until they become independent. So for this to happen, they have to bind with the mother, which is called as the filial imprinting. And if you look at mice and rats, when the young ones are given birth to, the young ones are in a very immature state. They will not have the development of fur, the skin will be naked, the eyes will be closed. And how these immature pups will recognize the mother is by the identification of the pheromone secreted by the mother. Mother releases the pheromone, and when the pups will lick on the secretions of the mother, they bind with her. These pheromones are called as imprinting pheromones. So these are the three types of pheromones what you come across in different animals. Apart from this, the pheromones are also classified as volatile and non-volatile pheromones. This is based on the molecular weight of the chemical substance. If the molecular weight is low, then you call it as volatile pheromone, and this is responsible for short time effect, like signaling. When there is a danger, it has to produce an alarm, which is a short time. It is required for a short time. So this is a short time effect pheromone. Or suppose some insect has to attract the opposite sex, then it will release the pheromone for attracting the opposite sex. So these short-term effect pheromones are having low molecular weight and they are called volatile pheromones. The second type is non-volatile, where you have the chemical substances which have high molecular weight, and this generally has long-lasting effect, which will be seen in the wild animals where they have to mark their territory. Generally, if you look at the animals living in the wild, they select a suitable, convenient place for them, which has got food resources and where they can guard their young ones. They identify such a place and they mark their territory with the help of the pheromone. Once they mark the territory, they do not allow other wild animals of its own species or any other species to enter into its home range. So this is done by the pheromones which are having high molecular weight, and you call it as non-volatile pheromones. So with this we are done with the introduction and different types of pheromones. Now we shall see some examples in insects as well as vertebrates. We shall start with insect pheromones. Lot of studies has been done in insects where people have deciphered many insects which are releasing pheromones, and if you look at the variety of glands present in the insect, you will be really fascinated to look at how the different cells are modified to become special glands to release the pheromones. We shall see some examples. If you look at the locust has got some epidermal cells which become specialized to release the pheromones, which acts on the nervous system. So if you look at the insect nervous system, there is a part of the brain called corpora allata. So the pheromone will act on the corpora allata, and what does this do? It brings about maturation of other members of the colony when they are. So these epidermal cells will release the pheromone which acts on the corpora allata, a part of the insect brain. And what does this do? This will bring about maturation of other individuals of the same species. It speeds up the process of reproductive maturity, and that's how the pheromone acts on other members of the same species. In Lepidoptera, where you have butterfly and moths, males have got a gland called a scent gland, and this scent gland releases the pheromone. And if you look at the wings have special glands which are called as androconia, and from this the pheromones will be released. And what does it do? It induces the process of mating. So it brings about mating between the opposite sex. Then we have Danus gillipus. Here the male will secrete the pheromone. It brushes the pheromone onto the body of the female with a small brush-like structure called as hair pencil. So with the help of the hair pencil, it brushes the pheromone on the female and brings about mating. In the Bombyx mori, the silk moth, female will produce a pheromone from the saculi lateralis. It secretes the pheromone bombicule, and the function of this bombicule is to attract the male. Another classical example is seen in the female emperor moth, scientifically called Endia pavonia. Here the female releases the pheromone to attract the male, and the male can sense the pheromone, and if the male is 13 kilometers away, even then it can sense the pheromone and it gets attracted and comes to the female to mate. So it is so strong. Bombicule is the pheromone secreted by Endia pavonia is so strong that it is capable of attracting the male, which is 13 kilometers away. So in this you can look at the different organs which are specialized to release the pheromone. In some insects there is a scaly patch, in some there is androconia, in some there are special glands like the Duffer gland, mandibular gland, and if you look at the ant, there are different types of glands present in the same individual, each one producing a different type of pheromone having a specific purpose. So you can just go through the diagram and you can read what are the specialized insect body part which are responsible for secreting the pheromone. Hymenoptera, where you have the honeybees, when we do social life, you will see that lot of the social life is controlled by the release of the pheromone. If you look at honeybees, in honeybees, they have mandibular glands. The queen and workers, both of them have mandibular glands which secrete a pheromone. They have studied the chemical nature of this pheromone and they have found out that it is 9-keto-decanoic acid. And what does this do? This will inhibit the ovarian development of the workers so that new queen will not be produced in the colony. The entire colony will have only one reproductively active female, the queen, and when she is there, no other females will mature, and the reproductive maturity of these workers is suppressed by the secretion of a pheromone which is known to be 9-ketodecanoic acid. Queen bee also secretes queen substance, and this queen substance will also suppress the development of the ovary so that new queen will not be formed. Queen bee again will produce another pheromone by the mandibular glands, and chemically it was identified as 9-oxodecanoic acid, and the function of this pheromone is to attract the male, which is called as the drone, and during mating or swarming flight, the queens and drones will go out for the purpose of mating. So the queen releases oxodecanoic acid to attract the drone, and then the mating will happen. If you look at ants, all of you must have seen how very well organized an ant colony is. If you look at the movement of the ants, they follow a path on which they are moving to and fro in search of the food. All this will happen because of the release of the pheromones. Ants will produce different types of pheromones for different purposes like mating, or finding the food source, or exploiting the food source, or recruiting the nest mates for the purpose of protection of the colony or to keep away the predators. So these are some of the functions for which the pheromones are released. In the ant, Iridomyrmex humilis, what they do is, when they find the source of food, they come back to the nest, and on the way back they leave a trail of pheromones, and other members of the colony will follow this path and find the food source. So they reach the food source with the help of the pheromone which is left as a trail. In Solenopsis, they have Pavon's gland. In Myrmica, they have poison gland. In Lasius, they have rectal gland. So in different ants, different glands are specialized to produce the pheromone, like Pavon's gland in Solenopsis, poison gland in Myrmica, rectal gland in Lasius. All of them will secrete a pheromone which is released like a trail to identify the location of the food. Leptothorax, here the workers will recruit specific number of the members. So if you look at how the ants are recruited for different purposes, let us take an example of an ant which is trying to get the food back to the nest. Sometimes you must have seen they have found a food source which is quite big than their own body, but some 2 or 3 ants are enough to roll that food to the nest. So when they find the food source, they will also give the information as to how many members are required to bring the food back. So this is done by the pheromones which are called as recruiting pheromones, which will also give the information about the food source as well as the number of individuals required to get the food source. Next, in Solenopsis, here you see that when a large food source is found, they release mass recruiting pheromone so that a group of ants will go and they try to get the food back. If the food source is quite big, many members of the colony will form a trail. Many members will go one behind the other to collect the food and bring it back to the nest.